everybody, to Zero to Biocomp, Biocompatibility 101 for MedTech professionals. I'm very pleased to be joined today by Michelle and Marina Daineko, and I'm going to ask them to introduce themselves. But before I do that, just a couple quick housekeeping announcements. Please use the chat vigorously during this event, as well as the Q&A. We're not promising that we get to all the questions during the call itself, but we'll follow up afterwards. This call is going to be recorded, and the recording will be made available along with the slides, the presenter slides, to everybody tomorrow in an email that will go out from Zoom. So there'll be links to that. We're going to provide some other links as well. And in the while we're getting started, I would like you all to please take a moment to answer this quick 3-question poll, which will help us frame the remarks appropriately during the call. And yes, that you get the idea. And let's have Michelle introduce herself first. Michelle.
Hi, I'm Michelle Lott with 𝗕𝗂𝗋. 𝗕𝗂𝗋 helps people, particularly startups and small to mid-sized businesses, navigate this challenging regulatory and quality environment. And we've helped over 100 companies now bring their products to market and keep them there.
That's wonderful. And Marina.
Hello, everyone. My name is Marina Daineko. I am biological evaluation consultant at Intrinsic Medical Group. So within Intrinsic Medical Group, we provide different services related to medical devices, such as biological evaluation, sterilization, cleaning, as well as reusable devices. So very niche, very specific, and I'm really happy to be there. So hopefully we would have some fun.
Wonderful. Thank you.
Thank you.
I am going to share my desktop, I think. So, okay. While I'm trying to bring up the agenda, let's go ahead and start with the first question. I'm going to end the poll, share the results. Are you seeing the results?
Mm-hmm.
Yeah. Wow.
So there you go. That's good information to have. And yes, so let's just go ahead and dive right in.
Sure. So we have a little bit of an action-packed agenda here today. So we have got a lot of questions that were submitted in advance. Be sure to put your questions in, because I'm going to be, as Marina is talking, I'm going to be kind of emceeing here and going back and forth between the chat and questions we've had submitted to kind of get us started. But you know, for those of you who follow either one of us, she and I have been talking about biocompatibility a lot on different platforms. I did a podcast recently with 𝗕𝗂𝗶𝗰𝗹 that Marina and I had discussed both beforehand and afterwards with the results of the discussion. And then shortly thereafter, Marina did a great presentation with the Medical Device Group. And both of them just generated a lot of conversation. So here we are to kind of keep that conversation going with some of the most popular questions. So Marina, you know, given that the title of this is from Zero to Biocompatibility, Biocompatibility 101, but with that said, it looked like about 42% of people were a little bit more confident than maybe a 101. Let's start with, like, what is biocompatibility, and like what are some of the nuances that the people who are at the lower end of that 40% versus the higher end of the 40%, like what are some nuances that both groups need to know?
Yeah, I really like this question, and thank you very much. And I'm really glad to see the voting here, like a little familiar or relatively familiar. So, you know, biological evaluation, biocompatibility, it's better to say, it's the property of the system. So we can't say like some material or some component is biocompatible. No, because we should know some specifics here. Like, for instance, not only the materials that are used for this specific component, but what is intended use of our medical device and some other aspects here. So I prepared actually really quickly the presentation. If you don't mind, I will share it because of
And just quickly, I said put the questions in the chat, but if you could actually use the Q&A, because that's a easier way to track it for after the webinar, because Marina will be preparing like a question and answer for all the questions we got that we may or may not have gotten to. So please use the Q&A.
Yeah, please, I like questions, just share them within the chat. Okay, I hope you can see my screen. Yeah, my presentation. So let's go. What's assessed within biocompatibility? Biocompatibility is applicable just to medical devices, and it's very important to understand and to use in our work. So first, we need to talk to regulatory as well as clinical to understand whether it's medical device, whether it's registered medical device. Then let's go into details. Not all the medical devices shall be assessed. We will talk about it a bit later, but within the standard, we have to assess the medical devices that come into direct or indirect contact with the human body. And why shall we assess it? Because of general understanding what's safety. Safety is freedom from unacceptable risks. And here we have to assess the potential for unacceptable adverse biological responses actually from this contact. So hopefully it's like more or less clear. And here just example, what's direct, what's indirect contact. Here you may see the medical device and it has direct contact, meaning physical contact with the patient. And here is the example of infusion set. And here I just marked the tube, and there is liquid that flows from the infusion bag through this tube. So tube doesn't have any physical contact with the patient, but liquid from this tube has. So it means, according to the standards, that the tube has indirect contact with the patient, and it means that the tube shall be assessed in our documents.
Yep.
And
So,
here,
Marina,
yep,
right there. One of the questions I got, you know, beforehand is like, what happens about those products? Because especially like in some of the 60601 series, they call out some biocompatibility requirements, and they say things like, you know, parts that are intended for direct or indirect contact with those biological tissues, etc., like you were talking about. But what about parts that are not, or might be not indirect, but I'm trying to
like
look
no
for
contact or like transient contact, some such
or
kind of
maybe
thing,
be
right?
like a lead inside electrode that doesn't actually touch the
Yeah,
patient. Like,
actually,
what are those parts that can
I
sabotage your biocompatibility?
actually, as I started this conversation, if we have categorized medical device, the device that is categorized as medical device and it's registered medical device, right? We need to perform biological evaluation in any case, even if it doesn't have any contact with the patient. What does it mean? It means that we just need to prepare the document that states like, okay, we have medical device, it doesn't have any contact with the patient. We don't need to perform biological evaluation on it, but we need to document this information. So it's required, it's a requirement to assess it in any way. So, and here actually very similar, oops, I'm sorry, very similar, nope, wrong. Yeah, here, example, case study, because I took it from the questions that were asked in advance. So here there is a question from medical electronic equipment. and standard 6061-1 actually. And the question is that there is like a specific clause that asks about ISO 10993 series of standards, and there is another clause that states only about risk management. And the question that we got is whether we have to do biological evaluation for such kind of devices. So if I can just second, yeah, show you these clauses, how they look like from the standard. So here one more time is about mostly risk management and applying risk management and parts that contact the patient, as well as very specific about biocompatibility of medical electrical equipment and systems. So first of all, we need to understand whether it's a registered medical device, whether it's a medical device itself. Then we need to understand whether we have, for instance, accessories, because there is a kind of Pandora box or tricky questions with accessories, because they can be able to be excluded from biological evaluation at all, because they can be not part of medical device and even do not influence on medical device functionality. And when they are excluded, we shall not do biological evaluation, for instance, for accessories. The example here you may see in a violet is a carrying bag for a pump, for instance. So it's
to clarify though, you mean excluded in the evaluation? Do you mean like in the actual testing, or in the analysis of the report about what you chose for testing and what you didn't and why?
Are actually excluded not only from the testing, but also excluded from biological evaluation, because it's not a medical device. And going back to the definition of when we shall perform biological evaluation, we have here the clause that it's for medical device. And when together with regulatory as well as clinical, this accessory is not part of medical device, is not like registered medical device, it means that we should not do biological evaluation on it. It's Pandora box. It's like, it depends, because accessories, we can have very different accessories that can actually impact the functionality of the medical device. But it's the beauty of biological evaluation to me, to be honest, because we have to consider very carefully our device. We need to know our device, and we need to cooperate with other colleagues from other departments here.
Okay. And somebody asked about unclassified devices. Where does that fit?
Unclassified? Unclassified devices. Do you mean unclassified as per ISO 10993 medical device regulation, or? Sorry, I didn't
We'll
get
get some
question.
clarification on that for after the webinar.
Yeah, please. It would be really helpful. So, talking about this example here, if we have medical device, we have to perform biological evaluation, even if it doesn't have any contact with the patient. We just need to document it. So we have the document that, okay, no contact with the patient. And surely follow ISO 14971 for risk management processes. Another case study that also was shared, and I really like it, was about cable assembly. And actually this cable assembly together with connector was shared with the laboratory. And what lab did actually, they grinded the entire assembly and put it into solvent, and predictably this test failed because of the presence of bromide in the contact pin. So what's the challenge here, or what was done wrong here? That we have to be careful, and we need to consider each element, each component, and identify what contact type it has with the user skin. For instance, here, these pins were designed in such way that they have no any contact with the patient. So we just need to, again, document it within our biological evaluation and prepare this sample for the lab properly, or describe this preparation properly. And here you may see, unfortunately, the manufacturer lost like 12,000 euros because they were forced to repeat this test, just removing these connector pins. And yeah, test passed and all good here. So that's why biological evaluation shall contain the information on not only like medical device and whole picture and intended use, but also we need to break into lines what components are here, what is the contact type with the patient for each component. Okay, if we can go next, here is a really cool example, and thank you very much for sharing this. So here we have measuring blood pressure device with a finger cuff. So here, this picture, I just took it from AliExpress, is just for illustrative purpose, how it can look like. So it's not about this particular specific device. So what was the challenge here? The challenge was that the cuff connects to a wrist-worn unit through a cable linked to a central device. And this wrist unit is secured with Velcro band. And because it's only to fixate the wrist part in vicinity of the fingers, it might as well be worn over a sleeve. But all the documentation, like instruction for use, all the marketing information, etc., they had a picture that the wrist unit being worn directly on bare skin. And Notified Body noticed it, and they asked actually about biological evaluation of this part. Unfortunately, tests that was performed to provide evidence that this wrist unit is biologically safe failed. So because it was not considered, it was not intended even, but all the information, marketing information, instruction for use, etc., had this picture that this wrist unit can be used on bare skin. And here we have the contact with intact skin, right? So, and here the numbers are really big, they're really huge, like 100,000 euro requires to revise all these materials, like together with translations, etc., etc. And what was done actually, it was like from business perspective, it was allocated 20,000 euros to be able to find new wristband materials that can be compliant with the requirements and can be claimed as biologically safe, just because not to rewrite all the papers. And here you may see on the list all the documents as well as processes that required update when it was found. So that's really impressive how different teams shall talk to each other, right? And understand how the device shall be used actually.
So this is probably a good time to talk about changes like both like maybe in your test plan or justifications that can be done maybe before a test takes place, or maybe after you got some maybe unexpected results and you realize that maybe you made some of these mistakes, you know, where are the guidance or the boundaries around how you can the justification process?
Yeah, absolutely. So I just want to take a step back, if you don't mind, and share with you when biocompatibility biological evaluation actually shall be performed. And here you may see number one is prototype phase, and you might think like it's something new, and you're absolutely right, because this assessing of biocompatibility on prototype phase was included in the current draft of ISO 10NS3-1. And I do believe on the very prototype phase, it's valuable and important information information can be derived. Why? Because we don't need to perform like whole biological evaluation. We can do biological assessment, if that makes sense, just to see whether we have any materials of concern, anything that can give us headache later, you know, and also some other points here like design verification, for sure, medical device change, emergent post-market information. And here I'm talking not only about complaints, but to our device, to our specific device, but also some information for applicable, for instance, for predicate devices or for devices from competitors, etc., as well as changes in BIOCOMP related standards and regulations. Like, for instance, we had recently -17 updated significantly, so we need to consider whether we need to remediate our biological evaluation reports or they are still applicable. And here, just really briefly, to medical device changes, so they are a lot. Actually, they can relate to design or material or packaging changes, changes in intended use, changes in patient population. Just imagine you have a medical device for the adults, and from business perspective, the team decided to extend their target population, for instance, for pediatrics as well. So it means that we need to consider toxicological limits as well as do our assessment to include this new group like pediatrics as well. Why? Because the weight is different. For adults, according to the standard, we have like 70 kilograms for adults, for men, sorry, and 58 kilograms for women. But pediatrics, we have like 13 kilos, so 3 kilo. Yeah, so here we need to take this into consideration. And there are some other changes like manufacturing and many, many others. And just a quick actual example, it's my favorite one, and I take it from the FDA guidance. They just have it within this guidance that, for instance, there is medical device and we are adding component, internal components that has no contact with the patient. What does it mean for us? If like follow the standard, we should not perform BIOCOMP, right? But there is a trick here, because to be able to apply this component during manufacturing process, the heat is applied to join to another component, and this another component has contact with the body. And from this perspective, we have to re-evaluate our biocompatibility to do biological assessment, just to understand whether there is any effect from this applying heat from this manufacturing process to cool our device. So that's very interesting. And if talking about changes, it's very important, and it's actually a common mistake when people are trying to assess something alone. So if even we have a look into ISO 14971, there would be a clause about cross-functional team, because we need to sit down all together and understand what is the impact, what exactly is the change about, and who else shall be involved there.
So Marina, we've got a good one, and that was on my list from before, but came up in the question and answer again. And I'm going to rephrase it a bit, because everybody in industry got a little excited about the FDA guidance that came out with the whole appendix of materials didn't require testing or whatnot. Can you speak a little bit about those exemptions and when they apply and can be used in justifications when you're first scheduling testing?
Absolutely. When talking about this exemption from FDA guidance, that's really cool thing. Why? Because we can just use this information and refer to this document, but there are surely we need to remember that this attachment is applicable to the components or materials that have contact with intact skin only. So not compromised, not like implant, but just intact skin. And there is like very specific list of materials within this attachment, right? But we still need to do biological evaluation from, for instance, literature review, right? Just to add the information, because the materials, especially polymers, can vary from one supplier to another supplier. But surely we can just refer additionally, not just, but additionally refer to FDA guidance saying like, okay, we have this material, it's like used for intact skin, it's used for devices that would be sold only in their states, because we are talking about the FDA, right? Here, this material is used for this type of device, also to show that, okay, it's applicable, it's kind of clinically established, and because it's included into the FDA guidance, so it can be considered as biologically safe. But also we need to take into consideration some other materials and possible manufacturing residuals and so on and so forth. So we need to see whole picture. We need to assess whether there are any additional risks there. For instance, from manufacturing standpoint, if we have cleaning, so for instance, residuals from cleaning. If we have, I don't know, some sterilization by ethylene oxide, then maybe we have residuals of ethylene oxide or ECH, you know, such kind of things shall be considered as well. But again, we can use it like back and forth, like all the time, and surely just put in, okay, this is from this attachment of the FDA guidance.
Yeah, and so it's fair to say that, you know, don't get too excited because something is in that list of chemicals or materials, because you still have to do a lot of work in just the justification and research and applicability to your product, you know, etc. and that process. And then something else that you brought up, hit home with me, is that, okay, yeah, if you're gonna only go into the US with your product, yeah, maybe that approach will work. But what about differences by country that you have to consider?
Yeah, I love this question. Thank you very much. Because in most cases, the companies are trying to have, and that's okay from business perspective, right? To have one biological evaluation for different countries, and that's absolutely okay. But here we have to consider the differences, like you just mentioned, right? We could have this exception for the states because of the FDA and the guidance, but Europe here and medical device regulation and ISO 10933-1 doesn't have such list. And okay, polypropylene is pretty inert. I saw some articles that call it like biocompatible, but I hate, you know, when it happens, because we have to consider medical device in its final finished form. So it means that we still have to consider manufacturing residuals, what's done with our polypropylene, and some other things. So so here I would say, if we consider like both the states, for instance, and Europe, we need to do comprehensive analysis in any case. I mean, from literature review, for instance, assessing manufacturing flow, understanding what is our device, collecting complaints if it's a clinically established medical device. But for Europe, for instance, we can't use these justifications that it is within FDA guidance. So we need to do all this evaluation and document it surely.
I think that that's also a good thing to at least touch on briefly, is that, you know, there's a big push in Europe to go to non-animal testing.
Yeah.
And everybody's like, when is the FDA gonna adopt non-animal testing? First off, speak a little bit to, I don't think it's that exciting, because I don't think it's gonna be any cheaper or quicker. And then B, the FDA is not nearly as excited about the rest of the world about this. So speak a little bit to that
Yeah,
dynamic going on?
absolutely. There is a big tendency within medical device regulation, and even with the current draft of ISO 10993-1 that right now is under vote, if I'm not mistaken. So to minimize animal testing and switch to 3R, like replace, reuse, replace, reuse. I'm like all the time forgetting the third one. Three R's principle or concept, right? So within our MDR, if we are applying here in Europe, so MDR, the regulatory bodies would not accept animal testing. And unfortunately, and there are some exceptions, like for instance, for sensitization, also for implantation, but there is like more and more shift to non-animal testing. And also one more concept that I can talk a bit here is about using real world evidence and information. So here we are talking about streamlining and structuring the data that is already available for some medical devices, because we can find predicate medical devices or equivalent or similar medical devices. So how they perform, what are clinical studies, what is the data about it. And using this data, leveraging this data, we can also make conclusions about biological safety. Because look, when I was talking to Dr. Hopkins, he actually made some time ago, quite a long ago, he made a very nice post about how can we use this test, this animal test, if their reliability is around like 60%, if I'm not mistaken, you know? So what about other 40%? So what to do here, right? Because some tests can be inappropriate because so big difference between what we have within the test lab and clinical conditions when actual patient is using this test. So that's why real world data and evidence are like very popular right now, if I can use this word. And I do believe that this data can be leveraged for our medical device. But when we are talking about the FDA, the case is a bit different, because I know that there are some discussions around it, and some people are trying to go within non-animal testing more and using also real world evidence, but some people are not so eager to jump to in vitro or in silico testing, actually. Yeah. So, and when we are talking about the FDA, they tend to use animal testing preferable to in vitro testing, just because of this approach. But again, I faced with different cases there, and it depends on the personality, I would say. But mainstream, yes, to use animal testing. So here, it's very important that we can share the information, that we can educate each other, that we can actually leverage the information we have. When we are applying, for instance, for both like the FDA and here in Europe, so I know that Europe can accept animal testing, because we can say like, okay, FDA is asking for it, or for instance, it was done for the FDA, like first entry to the market and then leveraged here for Europe. So it depends. Again, unfortunately, the answer is not like
so you
black
could
and
say
white.
it was already done, you know.
Yes, exactly,
what's done
exactly,
is done.
exactly.
Yeah.
Exactly. It was already done, for instance, for the FDA, and market it's sold on the American market, and then people want to bring it to European markets, and like,
so that might be one more justification for a
yes,
US market entry over an EU right now.
yes,
Interesting.
yes.
So here's another good one, and it's something that you and I have talked extensively about, and that almost the whole podcast I did with 𝗓𝗓 was about this. But how does one go about selecting a test lab, and what's going on politically right now with all the biocompatibility test labs? How do you know what a good one versus what, one on the naughty list is right now?
Oh yeah, it's very hot topic actually. Right now, and there are quite a lot of questions like this: how to pick the right lab and what to do with it, right? So maybe I do believe that it's not a secret that right now it's kind of ban from the FDA to perform tests in Chinese and Indian labs. So, but I have heard the information that the test can be performed in Chinese or Indian markets after FDA inspection actually. So FDA is going to inspect such labs, and surely this information would be available on their website. And when it's available on the website, like, okay, it's safe to use here. Yeah, so then
by on the FDA website, are you referring to the
ASCA?
Accreditation, yeah, ASCA
It's
accreditation
another
scheme
point.
for conformity assessment. Is that right?
It's another point, but the first point is that FDA is going to audit them. It's just recently used it.
So
I have heard
outside of
last
that
week.
process.
Yes,
Mhm.
yes. So regarding ASCA, it's actually second point to be 100% sure what to do right now before like all these audits, before like some other information is available, is just to go to the FDA's website to ask a program actually, and have a look, like, type in biocompatibility and see what labs are available for the testing regarding biological safety here. So, and from this standpoint, again, safe to test.
And I
Yes.
just want to point out to people that may not understand the ASCA program, just because a lab is on the list for ASCA, you've got to look for two things. First, that it's certified for its sampling methodology as a whole, because that's like step one, because if you can't, properly sample, then they won't qualify you for any of the tests. And that's also a basic quality system. But then the next level of that is not only do you have to have this kind of general quality system and sampling technique, you also have to have test specific inspections. So you got to look for, not only is the lab got on the ASCA list, but they're on the list for the tests that you're hiring them for. So that it's not just a carte blanche.
Yeah, that's a very valid point, and thank you for bringing it up, because when you are looking for the lab in this ASCA program on FDA website, it's actually very clear, like, okay, they can perform cytotoxicity, just example, such kind of thing. So it's very important to have a look into it. Yeah, very valid point. Thank you. I also know that there would be a discussion around ASCA program and -17 extractables and leachables in October or November, I don't remember. So it's open and free. So feel free to register, because I'm just waiting with popcorn, to be honest.
Is that with the FDA, like one of their town halls or open webinars?
Yes, but not like town hall where they just present, but rather open discussion actually.
Okay.
So
Perfect.
here we would,
yeah,
and maybe I will provide the link to their website where you can register, because here on this call we would be able to talk from like industry representatives actually, and really important, interesting topic. I will provide the link, and I do believe Sean can help us with distribution of this link to everyone. Yeah. So
so here's the other one that I see a couple of questions that are kind of related to this topic, and that's understanding contact duration. And I'm going to go beyond just the standard, 24 hours or less, 30 days or less or more, to like, let's start thinking about continuous use. So talk about a little bit about those two topics and how you have to think about them together.
You mean like contact duration, like, okay, all the time, right? And second, sorry.
Like if you say something that you need to replace it daily, and so like,
Yeah.
are you gonna do a 24-hour or less? Or if
I
they
see.
have to like put on the nasal cannula every day?
Yeah. I see what you're talking about. So actually, it's very common mistake that sometimes a cumulative effect is not considered. And not only within the classification of or categorization of our medical device and whole assessment in biological evaluation, but also duration of use during testing, because it's important, for instance, for toxicological actually risk assessment to understand how long our medical device can be used. Oh, I could see some hearts, some some ups. So cool, thank you. So, and here we need to work in collaboration with clinical affairs as well as regulatory affairs to understand whether it's like really can be used, I don't know, like altogether, I don't know, for years, right? Or their usage of this particular medical device can be limited to some, I don't know, to some standpoint. So, and surely when we have this cumulative effect for medical device, like lifelong, if that makes sense, right? So we need to go into very throughout biological evaluation. And for instance, for our type of medical device, this, for instance, chronic toxicity, let's consider, right? So it's not required for our type of medical device. But when we consider cumulative effect and consider like, okay, this is lifelong duration of use. So we need to do not only like literature review and assessing all the things that we are doing for each biological evaluation, right? But we also need to have a look into toxicological risk assessment and bring this contact duration here as well to be able to calculate the right limits. Yeah,
Okay.
hopefully, yeah, hopefully that helps.
So we talked about some of the case studies. What are some of the other common mistakes that you might have seen either in sample submission, test selection, the wrong type of test? Like, is there anything else that you feel like you need to leave us with some wisdom on here to?
Oh, there are quite a lot of common mistakes. Maybe the most common, I would say, it's absence of biological evaluation plan when you are going to the lab. Because lab, surely they can help, they can test whatever you like bring to them, but their core the core idea of biological evaluation is to understand medical device, to utilize the data we have for our medical device. I mean, for instance, from clinical, from post-market, if we have available post-market information, from a literature review, from available databases with toxicological profiles whatsoever. So what's biological evaluation plan about is just to structure the information to understand what's covered, what biological effect covered, what's not covered, and help to understand biological hazards, because we just can't go and test everything. We just need to understand, okay, when we test, what we are checking. So, here, that's why in ideal world, biological evaluation plan shall be signed off before you go to the lab, because what biological evaluation plan
There's a public
contains.
service announcement, people. All of your protocols and plans should be signed off on before you do the testing, regardless of biological evaluation or otherwise.
Yeah, it's very valid point, and thank you for highlighting it. And BEP also contains normally test strategy. Like, okay, we identified gaps. Okay, so how we are going to provide evidence, or what shall we do? And one of the input here, like what shall we do, it's actually to do the test, right? But here it's not too easy, like say, okay, CITA, we are done, you know, like 5 samples, that's it. No, there are different types of CITA, like what exact CITA we are going to use because of our device, like whether we have just the contact with intact skin or the implant. So we need to consider this and put all these details within our biological evaluation plan. Also, the quite common mistake is with extractables and leachables, like wrong solvents, for instance, are chosen, or wrong temperatures are chosen. Or for instance, we have adhesives, and oh my gosh, we have adhesives, and these adhesives can absorb some water or some other liquid, so it shall be considered as well. And all these details normally, yeah, please.
Share with the good people, like how much time and money average extractables and leachables cost.
A lot.
Yeah.
I mean, with the it's actually if it's not the most expensive, I would say this is the most expensive.
It's easily,
because
usually in it like a 6-figure sum, and it's like 8 to 10, sometimes 12 months.
Yeah, and why so long? Because usually the labs need to identify their basics. I mean, they are checking how it works, whether it works. In some cases, it doesn't work. So they are trying to understand or find other solvent or maybe mixture, you know, and all these things are taking time. Also here, there is a trick if you're talking about extractables and leachables. So leachables are more similar to clinical use, like actually what can leach from our medical device to our body. And extractables is about everything. It's kind of net that we are trying to get everything. And this analysis, like first of all, extraction, it takes time in different solvents, and then we need to do proper analysis to be able to identify what we actually found within our medical device. And here there is also a trick to a question how to choose the lab, because for the customers, I usually recommend to reach out to the lab that have experience working with similar devices. Why? Because they can have good libraries to be able to identify these chemicals that are pretty important to toxicological risk assessment, to extractables and leachables, chemical characterization, I mean here. So
that's important.
Yep.
To link back to our discussion about how to select a lab. That's an important question, is to ask them specifically about your device type and their experience, again, test by test with your device.
Yep. That's really, because I would even recommend to ask all possible questions, not only about certifications or checking them, but also asking for showing their protocol templates, you know, and absolutely asking about the devices, whether they have experience working in. It's important. One more thing that maybe I would like to highlight, our final thing here, is about it's about expertise requirement as per ISO 10993-1, that here we can have our education, here we can have our experience at work, but also taking part in different conferences, summits, networking with people, and not doing biological evaluation alone, but rather with a talented team to be able to do proper evaluation. So unfortunately, it's also quite a common mistake just to do everything alone. But yeah, we just need to talk, we need to share the information, and we need to think loudly and discuss the things with other professionals. I do believe that this soft skill of communication is our must to be able to find the information, share the information, and make some conclusions actually here.
Mm-hmm. What about, are there any trends that we haven't talked about that either from things that you've seen in active review being challenged by either the FDA or their notified bodies, or what do you see coming for the future?
Oh, just a short case actually, because I really like it. The company submitted a technical file here in Europe, and notified body, it's actually about surgical instrument, and notified body said like, okay, you have stainless steel, please provide justification about 𝗕𝗂𝗾𝗊. And the beauty here is that surgical, according to the technical specifications, there is no any cobalt in this stainless steel, you know, but cobalt is residual and it goes directly together with iron no matter what. So you will find cobalt if you have iron, any iron material like stainless steel, for instance, you know. And what they actually asked and what I did, I provided justification about this very small amount of cobalt within biological evaluation. So be careful here. And here I'm talking actually about carcinogenic, mutagenic, toxic to reproduction, as well as endocrine disruptors, all the things that are mentioned within medical device regulation. And it's actually main difference from what we have within the FDA, because FDA doesn't have such lists.
Yep.
Yeah.
Which kind of sums up, like, I think several of these questions that I have seen come through about, I'm just going to rattle off a couple of them and then make a point about it. Like, are biocompatibility studies strictly supposed to be performed for finished products, or the material studies fine to show in a 510K? If my supplier does not mention the grade of raw material, what should I do to demonstrate? So on and so forth. We've got several kind of a little bit more specific questions about that. But I think the answer to all of those, and kind of a point of a lot of the questions we've talked about, is that it comes back to that biological evaluation plan, and that it is comprehensive, that it's done in advance. And if you have any hope of being able to justify doing or not doing, or doing a smaller subset of testing, it is going to be from a very thorough, well-researched, and well-documented biological evaluation plan.
Yeah, absolutely. And I would say that in some cases, because usually biological evaluation plan, again, contains the information about sample, how the sample was chosen, why this exact sample, you know, with justification, like whether it's worst case or whatsoever. So in some cases, this can be justified, but as you said, everything shall be documented within biological evaluation plan. Not only like, okay, we picked this one because something is not provided or something not done, but no, it shall be a scientifically valid justification why we can use and why we don't see any additional risks while using this exact sample to testing. But really very shortly, if you don't mind, about future. So here I could see several streams actually. One of the stream is using AI for biological evaluations that can help to streamline the information, but I don't think it would replace us, because we need to see like whole picture, the dependencies, etc., rather like help us there. Yeah.
Yeah, it might streamline but not replace.
Yeah, streamline, helping to accumulate the information, because I like the approach when we can reuse some specific information about materials that are used for different medical devices, but used for us, for our medical device, for instance. Right, what else can I name here? So surely going into in vitro testing mostly, and reusing already existing data, for instance, from clinical evaluations. And here I'm talking about real world evidence that is pretty popular right now. And I hope that in future, when we have this -1 published, hopefully this year, in the end of this year, right, we would be able to identify some concerns in the very beginning, like on prototype phase. So it would help us to save a lot of money and time, because we would know, hopefully, some concerns in the very beginning, and we would be able to do something with this, like to implement some control measures.
Yeah. Well, we're almost out of time here. There are so I think this is relevant. Luke just asked who was an early contributor to our questions here. Do consulting firms exist that can specifically advise on biocompatibility plans throughout the design phases, like on what is and not stupid to design? I think the answer to that is yes, Luke. Both IMG and Lean RAQA can help you sort out the stupidity.
Yeah, I just wanted to add really quickly here that on the design phases, like if you're talking about prototype, for instance, not design verification, we can use biological assessment, not like plan, very deep with the testing, but rather assessing together with cross-functional team, yeah, on the very beginning. But yeah, both Lean RAQA as well as IMG can definitely help.
Mhm.
I would like to take a moment to thank both of the speakers for joining us today. This has been very, very informative. Thank you very much. I would like to remind everybody to take the survey, which is going to pop up on your screen when you leave the call. Your feedback is very important to us. I've put a number of links in the chat. I recommend that you make use of those links. If you would like to connect with either Marina or Michelle on LinkedIn, please do so. And if you have any questions, please email me, Sean@virtub2b.com, which again is in the thing. If you haven't requested your certificate of participation, or attendance, please put your name as you would like it to appear, along with your email address in the chat, and that will be sent to you in the next couple days. We have to do this manually, so please be patient. Again, there are 2 upcoming webinars. Our first Spanish language webinar, which is Health Tech LATAM, is going to be really exciting about getting your 510K, and then clinical data requirements EU versus US. Both really exciting coming up this month. Please check those out. And remember, subscribe to medtechleadingvoice.com and get all of the latest from experts like Michelle and Marina. Thank you both very much. Any closing words or thoughts?
BEP.
Yeah, just remember this.
Yep. If there's anything you take, it's do a biological evaluation plan. To Luke's point, early and often.
And if you have some additional questions that you were too shy to put into the chat, please feel free to reach out to any of us. We would be more than happy to address the question and to help, but surely on high level because of the NDA.
Wonderful. Thank you very, very much, both of you. I hope everybody has a great day and a great weekend.
Thank you. Likewise. Enjoy your day. Bye-bye.